Cofactor, Wikipedia audio. A cofactor is a non-protein chemical compound or metallic ion that is required for an enzyme's activity. Cofactors can be considered helper molecules that assist in biochemical transformations. The rates at which these happen are characterized by enzyme kinetics. Cofactors can be subclassified as either inorganic ions or complex organic molecules called coenzymes, the latter of which is mostly derived from vitamins and other organic essential nutrients in small amounts. A coenzyme that is tightly or even covalently bound is termed a prosthetic group. Co-substrates are transiently bound to the protein and will be released at some point, then get back in. The prosthetic groups, on the other hand, are bound permanently to the protein. Both of them have the same function, which is to facilitate the reaction of enzymes and protein. Additionally, some sources also limit the use of the term cofactor to inorganic substances. An inactive enzyme without the cofactor is called an apoenzyme, while the complete enzyme with cofactor is called a holoenzyme. Some enzymes or enzyme complexes require several cofactors. For example, the multi-enzyme complex pyruvate dehydrogenase at the junction of glycolysis and the citric acid cycle requires 5 organic cofactors and 1 metal ion, loosely bound thiamine pyrophosphate, covalently bound lipoamide and flavin adenine dinucleotide, and the co-substrates nicotinamide adenine dinucleotide and coenzyme A, and a metal ion. Classification. Organic cofactors are often vitamins or made from vitamins. Many contain the nucleotide adenosine monophosphate as part of their structures, such as ADP, coenzyme A, FAD, and NAD+. This common structure may reflect a common evolutionary origin as part of ribozymes in an ancient RNA world. It has been suggested that the AMP part of the molecule can be considered to be a kind of handle by which the enzyme can grasp the coenzyme to switch it between different catalytic centers. Cofactors can be divided into 2 major groups: organic cofactors, such as flavin or heme, and inorganic cofactors, such as the metal ions Mg2+, Cu+, Mn2+, or iron-sulfur clusters. Organic cofactors are sometimes further divided into coenzymes and prosthetic groups. The term coenzyme refers specifically to enzymes and, as such, to the functional properties of a protein. On the other hand, prosthetic group emphasizes the nature of the binding of a cofactor to a protein and, thus, refers to a structural property. Different sources give slightly different definitions of coenzymes, cofactors, and prosthetic groups. Some consider tightly bound organic molecules as prosthetic groups and not as coenzymes, while others define all non-protein organic molecules needed for enzyme activity as coenzymes, and classify those that are tightly bound as coenzyme prosthetic groups. These terms are often used loosely. A 1980 letter in Trends in Biochemistry Sciences noted the confusion in the literature and the essentially arbitrary distinction made between prosthetic groups and coenzymes group and proposed the following scheme. Here, cofactors were defined as an additional substance apart from protein and substrate that is required for enzyme activity, and a prosthetic group as a substance that undergoes its whole catalytic cycle attached to a single enzyme molecule. However, the author could not arrive at a single all-encompassing definition of a coenzyme and proposed that this term be dropped from use in the literature. Metal ions are common cofactors. The study of these cofactors falls under the area of bioinorganic chemistry. In nutrition, the list of essential trace elements reflects their role as cofactors. In humans, this list commonly includes iron, magnesium, manganese, cobalt, copper, zinc, and molybdenum. Although chromium deficiency causes impaired glucose tolerance, no human enzyme that uses this metal as a cofactor has been identified. Iodine is also an essential trace element, but this element is used as part of the structure of thyroid hormones rather than as an enzyme cofactor. Calcium is another special case, in that it is required as a component of the human diet, and it is needed for the full activity of many enzymes, such as nitric oxide synthase, protein phosphatases, and adenylate kinase, but calcium activates these enzymes in allosteric regulation, often binding to these enzymes in a complex with calmodulin. Calcium is, therefore, a cell signaling molecule, and not usually considered a cofactor of the enzymes it regulates. Other organisms require additional metals as enzyme cofactors, such as vanadium in the nitrogenase of the nitrogen-fixing bacteria of the genus Azotobacter, tungsten in the aldehyde ferredoxin oxidoreductase of the thermophilic archaean Pyrococcus furiosus, and even cadmium in the carbonic anhydrase from the marine diatom Thalassiosira weissflagii. In many cases, the cofactor includes both an inorganic and organic component. One diverse set of examples is the heme proteins, which consist of a porphyrin ring coordinated to iron. Iron-sulfur clusters are complexes of iron and sulfur atoms held within proteins by cysteine residues. They play both structural and functional roles, including electron transfer, redox sensing, and as structural modules. Organic cofactors are small organic molecules that can be either loosely or tightly bound to the enzyme and directly participate in the reaction. In the latter case, when it is difficult to remove without denaturing the enzyme, it can be called a prosthetic group. It is important to emphasize that there is no sharp division between loosely and tightly bound cofactors. Indeed, many such as NAD can be tightly bound in some enzymes, while it is loosely bound in others. Another example is thiamine pyrophosphate, which is tightly bound in transketolase or pyruvate decarboxylase, while it is less tightly bound in pyruvate dehydrogenase. Other coenzymes, flavin adenine dinucleotide, biotin, and lipoamide, for instance, are tightly bound. Tightly bound cofactors are, in general, regenerated during the same reaction cycle, while loosely bound cofactors can be regenerated in a subsequent reaction catalyzed by a different enzyme. In the latter case, the cofactor can also be considered a substrate or co-substrate. Inorganic vitamins can serve as precursors to many organic cofactors or as coenzymes themselves. However, vitamins do have other functions in the body. Many organic cofactors also contain a nucleotide, such as the electron carriers NAD and FAD, and coenzyme A, which carries acyl groups. Most of these cofactors are found in a huge variety of species, and some are universal to all forms of life. An exception to this wide distribution is a group of unique cofactors that evolved in methanogens, which are restricted to this group of archaea. Metabolism involves a vast array of chemical reactions, but most fall under a few basic types of reactions that involve the transfer of functional groups. This common chemistry allows cells to use a small set of metabolic intermediates to carry chemical groups between different reactions. These group transfer intermediates are the loosely bound organic cofactors, often called coenzymes. Each class of group transfer reaction is carried out by a particular cofactor, which is the substrate for a set of enzymes that produce it, and a set of enzymes that consume it. An example of this are the dehydrogenases that use nicotinamide adenine dinucleotide as a cofactor. Here, hundreds of separate types of enzymes remove electrons from their substrates and reduce NADN to NADH. This reduced cofactor is then a substrate for any of the reductases in the cell that require electrons to reduce their substrates. Therefore, these cofactors are continuously recycled as part of metabolism. As an example, the total quantity of ADP in the human body is about 0.1 mole. This ADP is constantly being broken down into ADP, and then converted back into ADP. Thus, at any given time, the total amount of ADP and ADP remains fairly constant. The energy used by human cells requires the hydrolysis of 100 to 150 moles of ADP daily, which is around 50 to 75 kilograms. In typical situations, humans use up their body weight of ADP over the course of the day. This means that each ADP molecule is recycled 1000 to 1500 times daily. Organic cofactors, such as ADP and NADH, are present in all known forms of life and form a core part of metabolism. Such universal conservation indicates that these molecules evolved very early in the development of living things. At least some of the current set of cofactors may, therefore, have been present in the last universal ancestor, which lived about 4 billion years ago. Organic cofactors may have been present even earlier in the history of life on Earth. The nucleotide adenosine is present in cofactors that catalyze many basic metabolic reactions such as methyl, acyl, and phosphoryl group transfer, as well as redox reactions. This ubiquitous chemical scaffold has, therefore, been proposed to be a remnant of the RNA world, with early ribozymes evolving to bind a restricted set of nucleotides and related compounds. Adenosine-based cofactors are thought to have acted as interchangeable adapters that allowed enzymes and ribozymes to bind new cofactors through small modifications in existing adenosine binding domains, which had originally evolved to bind a different cofactor. This process of adapting a pre-evolved structure for a novel use is known as exaptation. A computational method, IPRO, recently predicted mutations that experimentally switched the cofactor specificity of Candida boydonii xylose reductase from NADF to NADH. Metal ions. Iron-sulfur clusters. The first organic cofactor to be discovered was NAD+, which was identified by Arthur Harden and William Yund in 1906. They noticed that adding boiled and filtered yeast extract greatly accelerated alcoholic fermentation in unboiled yeast extracts. They called the unidentified factor responsible for this effect a coferment. Through a long and difficult purification from yeast extracts, this heat-stable factor was identified as a nucleotide sugar phosphate by Hans von Euler-Chelpin. Other cofactors were identified throughout the early 20th century, with ADP being isolated in 1929 by Carl Lohmann, and coenzyme A being discovered in 1945 by Fritz Albert Lippmann. Organic. Vitamins and derivatives. Non-vitamins. Cofactors as metabolic intermediates. Evolution. The functions of these molecules were at first mysterious, but, in 1936, Otto Heinrich Warburg identified the function of NAD and in hydride transfer. This discovery was followed in the early 1940s by the work of Hermann Kalkar, who established the link between the oxidation of sugars and the generation of ADP. This confirmed the central role of ADP in energy transfer that had been proposed by Fritz Albert Lippmann in 1941. Later, in 1949, Morris Friedkin and Albert L. Leninger proved that NAD and linked metabolic pathways such as the citric acid cycle and the synthesis of ADP. In a number of enzymes, the moiety that acts as a cofactor is formed by post-translational modification of a part of the protein sequence. This often replaces the need for an external binding factor, such as a metal ion, for protein function. Potential modifications could be oxidation of aromatic residues, binding between residues, cleavage, or ring forming. These alterations are distinct from other post-translation protein modifications, such as phosphorylation, methylation, or glycosylation, in that the amino acids typically acquire new functions. This increases the functionality of the protein. Unmodified amino acids are typically limited to acid-base reactions, and the alteration of resides can give the protein electrophilic sites or the ability to stabilize free radicals. Examples of cofactor production include tryptophan-tryptophyllquinone, derived from two tryptophan side chains, and 4-methylidene-imidazole 5-1, derived from an allosteric GLY motif. Characterization of protein-derived cofactors is conducted using X-ray crystallography and mass spectroscopy. Structural data is necessary because sequencing does not readily identify the altered sites. The term is used in other areas of biology to refer more broadly to non-protein molecules that either activate, inhibit, or are required for the protein to function. For example, ligands such as hormones that bind to and activate receptor proteins are termed cofactors or coactivators, whereas molecules that inhibit receptor proteins are termed corepressors. One such example is the G-protein-coupled receptor family of receptors, which are frequently found in sensory neurons. Ligand binding to the receptors activates the G-protein, which then activates an enzyme to activate the effector. In order to avoid confusion, it has been suggested that such proteins that have ligand-binding mediated activation or repression be referred to as coregulators. History. Protein-derived cofactors. Non-enzymatic cofactors.